All right, well, hello everyone. My name is Leon Gua. I'm a PhD candidate at UMass Amherst, and I'm very excited to be here today to share with you, on behalf of my co-authors, our research comparing upper thermal limits in Anadromous juvenile river herring. If you're not familiar with river herring, they comprise two closely related species, Blueback herring and Alewife, both of which are pictured on the right. These two species are anadromous and distributed along the east coast of the US. And essentially every one of their life stages, they are incredibly important forage for a wide range of different species, some of which are pictured there on the left. Because of their, the populations remain depleted coast-wide due to a range of reasons, including predation, habitat fragmentation, fishing, and climate change. Because of their critical importance for recreational and commercial fisheries, they are managed currently in a unit with other shads. However, it's very challenging to manage them because, as you can see from this, even just this image here, they are morphologically very similar and they have similar life histories as well. So trying to have any sort of management strategies which really focuses on one species can be very challenging. However, as we're trying to move now toward climate-ready or climate-adaptive management, the question remains whether both species are going to be similarly affected by climate change. There are a couple of observations that have been made which may suggest that the two species might not have exactly the same climate resilience. In this map on the left, you can see the geographic distribution of blueback herring in gray and alewife in light blue. Generally, alewife are more northerly distributed than blueback herring, which means that they're associated with colder waters. Additionally, trawl surveys have shown that the adults are disappearing at a faster rate from the southern part of their range for alewife when compared to blueback herring, suggesting that they're undergoing a faster rate of northward climate range shift. Additionally, alewife have an earlier migratory phenology, so they enter freshwater systems several weeks earlier in the spring. This means that on average, they are associated with colder water spawning temperatures. And so generally, I think many people think of them as more cold tolerant than blueback herring. So collectively, these observations kind of beg the question, are alewife more thermally sensitive than blueback herring, and should we be managing their populations more conservatively in response? This question is very difficult to answer because there actually has not been a direct species comparison done for any life stage of the two species. We were interested in doing this direct comparison, but focusing in specifically on the juvenile life stage. This is because freshwater systems are generally susceptible to more extreme and variable thermal regimes, especially under climate change. Furthermore, we know that small differences in mortality and growth rates can accumulate to affect recruitment. So if we can really detect any sort of differences in thermal sensitivity at these early life stages, it might give us a better sense of what will happen with population resilience in the long term. So we set forth to create a study where we could actually compare upper thermal limits and thermal performance between these two species when they're acclimated to a similar environment. To do this, we went and collected juvenile river herring from what we considered to be about the mid-range, which was, in this case, Connecticut in southern New England. We collected blueback herring from Wethersfield Cove and alewife from Bride Lake and Dodge Pond. These sites were selected because they are historically known to be predominated by those specific species, and so we hope to increase our chances that we would only get those species for our experiments. We performed 2 paired collections in July and August and attempted as best as possible to achieve size overlap between the two species. This proved to be quite difficult this past year, but I made sure to incorporate size as a potential explanatory factor when I was interpreting my data. So I'll talk more about that later on. In order to test the upper thermal limits, we used 2 different experiments. The first was critical thermal maximum, or CT max. You can think of this as a test which tests tolerance to rapid increases in temperature. So for example, changes on a daily basis or when fish are switching between habitats. After collecting the fish, we train them on dry food and then place them into replicate tanks for acclimation for 2 weeks to either 24 degrees Celsius or 28 degrees Celsius. After the acclimation period, we then subjected them to the CT max test. In this test, the fish are acclimated overnight in the testing tank, and then we rapidly increase the temperature 2 degrees Celsius per hour. The temperature at which the fish are no longer able to remain upright is recorded as their CT max. This is because we would consider that if fish were to not be able to remain upright in the field, they probably would be predated upon or have difficulty breathing, etc. The other experiment we did was an acclimated upper lethal limit test, or an L50. This test instead measures tolerance to slower increases in temperature, so something more on the scale of a seasonal increase in temperature. The setup for this experiment was very similar to the previous one, where we acclimated the fish only to 24 degrees Celsius for 2 weeks in replicate tanks. After this point, we began to slowly increase the temperature 1 degree every 2 days. This slow increase of temperature does allow the fish time to acclimate, and so this can generally be considered a test which is more ecologically relevant for wild populations. Additionally, we fed the fish their maximum food ration every day and recorded that amount as a measure of feeding performance. The temperature, which caused 50% mortality, was recorded as the L50. So the hypothesis that we have is that alewife juveniles will be less warm tolerant than blueback herring, demonstrated by a lower critical thermal maximum regardless of their acclimation temperature, lower upper lethal limits, and lower upper feeding limits. So let's go ahead and see if our hypotheses were true. This first graph I'm showing you is from the CT Max experiment, where we rapidly increased the temperature 2 degrees an hour. On the x-axis are the 2 acclimation temperatures we used, and on the y-axis is the critical thermal maximum, or the temperature at which fish could no longer remain upright. For this graph and every graph after this, the 2 species are going to be differentiated by their shape and color. So alewife will be orange and a circle, and bluebacks will be blue triangles. I've also included here some p-values from a generalized linear model I ran on this data. The first thing you'll notice is that there is no difference in the CT max between the 2 species, regardless of acclimation temperature. This was very surprising to us, as we actually did expect alewife to have lower CT maxes than blueback herring. Additionally, we find that both species are able to increase their CT max when they're acclimated to a higher temperature. This suggests that both species have some thermal plasticity and are able to increase their thermal tolerance in reaction to their environment. Finally, we did find that there was no difference in the CT max with mass, so at least for this measure, we see that there's not an effect of juvenile size on their thermal tolerance. Now we're going to move into the results from the slower increase in temperature study, the acclimated lethal limit. So the x-axis here has the different temperatures that the fish were in. The y-axis has cumulative mortality. I marked the 50% mortality line with that red dash line there. The first thing that you can see that, again, across all of the different temperatures, we really see no difference in mortality between the 2 species, and this matches what we saw in the CT Max test. Furthermore, we find that these species have incredibly high survival rates right up until they reach their lethal limit, and it suggests that, at least for survival, they have a very broad tolerance.
9 minutes.
Thanks, Chris. Finally, we see that the lethal limit is very similar to the CT max we measured for fish acclimated to the same temperature. This is not very common for fish species, as usually when you do CT max tests and you have that really fast increase in temperature, you often overestimate what fish are actually able to survive in the wild. And so to find that these measures are so similar for these 2 species is very surprising. This last graph, and now we're turning toward feeding performance. So again, on the x-axis, we have the different temperatures, and now on the y-axis, I have the percent of the food ration that was consumed at each temperature relative to the absolute maximum ration that was ever consumed for a given tank. In this case, I'm really going to focus on the upper part of this curve, since we're really talking about upper feeding limits. We see at 31 to 32 degrees peak food consumption for the 2 species. and significant drops in feeding performance thereafter. Generally, when you see these declines, the fish are actually decreasing how much food they can eat, and I think it's still debated, why exactly these fish are not able to eat as much as they are, but of course, declines in feeding performance have significant implications when you're in the field. And finally, we find that no food was consumed at 35 degrees Celsius. I ran a generalized linear model just for the upper part of this graph. The p-values are there on the graph as well. And essentially, we find that above max consumption, there was no significant difference in the feeding performance between the two species, again, matching what we found in the other experiments. In this particular case, we do find a significant effect of mass, where larger juveniles were eating proportionately less of their body mass. Again, this is not a surprising finding, as this is true for most fish species, but it does mean that moving forward, we need to be cautious when we're interpreting this feeding performance results, as we might have some confounding factors between size and species. Finally, we saw that drops in feeding performance occurred only 2 degrees below the lethal limits. This is not very common, at least for a species we studied in the lab, like brook trout. Usually drops in feeding performance will occur something like 6 degrees below lethal limit, and fish will actually stop feeding several degrees below their lethal limit, whereas for blueback herring and alewife, it seems all of that is really truncated within a couple of degrees, and they're able to keep feeding right up until they're basically dropping dead. So it, again, showing perhaps some very broad performance and tolerance across temperatures. All right, so now I'm going to recap and talk about some implications from this study. First, we find that there's no significant difference between juvenile upper thermal maximum, lethal limit, or feeding performance. And so we have to reject our hypotheses that alewife are less warm tolerant than blueback herring. It's possible that just based on the particular experiments we did or the performances we measured, we were not able to detect differences. But if we had measured, for instance, growth rate, there may have been some difference between the two species, and we just simply did not detect it within this study. Alternatively, it could be other life stages like the egg, larval, or adult life stages where differences in thermal sensitivity occur. Alternatively, if continued studies on this subject find that there actually is no difference between the two species in their thermal tolerance and performance, those differences that have been observed in distribution or phenology could be due to other biotic factors. There could be things like competitive exclusion between these two species or other species, or even just differences in how they're utilizing habitat and prey. All of these things are, of course, important for management to understand what are the really significant factors contributing to changes in the population dynamics in the future.
Less than 5 minutes.
Thanks, Chris. The second major finding we had was finding broad thermal tolerance and performance for both species. This likely reflects their broad and overlapping geographic distribution. However, we do see, at least with the measures we had, that these fish are surviving and performing fairly well right up until they're reaching their upper thermal limit. So for management agencies who are monitoring juvenile populations of river herring, if you're only using presence, absence, or abundance data, it may be very difficult to foresee declines in the populations related to temperature. And in my previous studies, I've found that growth rate, for example, will decline beginning at 24 degrees and just continue to decline. And so it may be important to be measuring things like growth rate in addition to abundance data to really make sure you're having a good gauge of population health. With that, I have many people to thank from my colleagues at UMass Amherst, Yale, UConn, Fish and Wildlife Service, USGS, CT Deep, and our funding sources. I also would like to formally apologize to Eric for not having a super interactive component of my talk, but I will share my blueback herring pumpkin as my creative aspect for those of you who can actually see it. Happy to share more upon request, and I am happy to take any questions. Thank you all for listening today.